First of all, thank you very much, Munir and others for inviting me to speak here. I hope you'll be gentle with me. I'm an epidemiologist, not a geneticist when it comes to questions. And in fact, when I was asked in 2005 if I would take on leading UK Biobank, one of my first actions was to persuade people who were very critical of the study that it wasn't a genetic study at all, it was just normal epidemiology. And then people said, oh, if it's not genetics, that's fine, you know, we're not worried about it. We kind of got in there through stealth. And about 5 years ago, when the funders did the review and asked in the review panel meeting what had UK Biobank delivered that it was set up to deliver, my one-word answer was nothing. There was a kind of pause at that point. But that's true, because with a prospective cohort, you really need time. You need to let it mature. And it's really in the last 5 years that it has started to mature. But it really is the outcome of the vision of the MRC, now part of UKRI, and the Wellcome Trust in setting this up in the first place in the UK, way before anybody else thought about the value of having a large prospective cohort for looking at not just genetics, but lifestyle, environment, on a whole range of diseases. And now the British Heart Foundation, Cancer Research UK are part of that vision and the support. So what did I have to do at the beginning? Well, the first thing was to explain why prospective cohorts are so valuable. Back in 2005, people were doing case control studies of genetics. It was a very efficient way. You get people with disease, you get people without disease, you compare their genetics, and you get answers quickly. And of course, that's the Wellcome Trust Case Control Consortium, which was the first study showing the value of GWAS. But if you're interested in all sorts of other risk factors that are changed by disease, I mean, if you think about it, what's the best way to get people to stop smoking? Encourage them to have a heart attack. Then they stop smoking. But you get reverse causality through doing that. And so if you're interested in all of the different determinants of disease, socioeconomic, environmental, lifestyle, and genetics, you need to study people before the disease changes their risk factors. And the beauty of a prospective cohort is that despite the disadvantage of having to wait, is it becomes a resource to do everything about everything. You can look at a single risk factor on many diseases. Think of Richard Doll and the British Doctors study showing that tobacco not only caused lung cancer, but many cancers, lung disease, heart disease, etc. But also you can look at the effects of many risk factors on a single disease. So as a resource, it's a fantastically cost-effective approach. But the problem, not only about duration, but also is about scale. Because inconveniently, only a small proportion of the participants in a particular cohort will develop any one particular disease. And it's the numbers of cases of a particular disease which will determine your power to assess associations. And so then the question is, well, how big is big? And people ask, why was UK Biobank 500,000 people? And the answer is because there wasn't enough money for 1 million. I mean, it's a completely arbitrary number. And of course, David Hunter will be telling you about how it should be 5 million. And you'll get more information by bigger scale. But to kind of illustrate why 500,000 kind of makes sense, we looked at data that Sarah Lewington in Oxford had pulled together from old prospective cohorts from around the world, looking at 2 strong risk factors, age and blood pressure, on a common condition, coronary artery disease mortality. And out of those 1 million people, I said, randomly select 5,000, a Framingham unit of epidemiology, and look at the association between blood pressure and coronary artery disease mortality at different ages and different blood pressure levels. And as you can see on this graph, you can't see very much. There's a kind of cloud with wide confidence intervals around where the point estimates are. You go to 50,000, a British Doctors unit of epidemiology measure, and things start to come into focus. But there's been a long debate when people were looking at studies of this size. Is higher blood pressure not associated with higher risk in older people? In which case, Is it worthwhile lowering their blood pressure? Is there a J-shaped curve? Younger people, whereby lowering blood pressure increases risk? Or is it chance? So you get up to 500,000 people, it's like a microscope, everything starts to come into focus. You get these beautiful log linear associations. Now, I say log linear because everybody's had a 2-year training in epidemiology in the last couple of years. We're used to using log scales now, and you can see a log scale here, straight line association, flatter at higher age than lower age, indicating that in relative terms, it's weaker. But because it's a log scale, the absolute differences for any given blood pressure difference are bigger at older ages. So in absolute terms, the interaction is stronger at older ages. But you can see where you get scale, you can look at interactions, you can understand those interactions and the shape of the association. But these are 2 strong risk factors for a common condition. So you can see that 500,000 is probably not enough for many things we want to study. So UK Biobank was set up. 500,000 people from around England, Scotland, and Wales were recruited. We located the recruitment centres in places that would give us quite a lot of heterogeneity. So if you go to Scotland, very difficult to get ethnic minorities, but if you go to Croydon or to some Midland towns, then you can get larger proportions. There's still minority in the cohort, but still valuable to include them, and as many of them as you can. Rural, urban, and different socioeconomic status. Very detailed assessment in terms of various exposures from questionnaires, from physical measurements, and from biological samples. And people always go on, quite rightly, about the altruism of 500,000 participants that provide all of this information with no feedback of individual results to them. But I think it's also worth bearing in mind that without the altruism of the UK medical research community, there wouldn't be UK Biobank. An enormous amount of work went into determining what questions to ask, how to ask them, what measurements to make, what biological samples to collect, doing detailed pilots to ensure that we collected samples that would allow assays to be done 15, 20 years later that couldn't be done at that time. So there was a huge amount of input from across the UK in terms of research community as well as the participants. And consent to access to all their medical and other health-related records, including things like socioeconomic measures, and for recontact of participants for further assessments, but not involving feedback of individual results to them, which is a constraint. So when they talk about the kind of unique combination of 4 dimensions, the scale is obvious. But I think the other important thing is the depth. So very detailed information about lifestyle, environment, and medical history, supplemented by linkage into the health records, but now increasingly by assays. So the first thing that was done in the laboratories in Stockport, where the coordinating centre for UK Biobank is, was a set of biochemical measures. The hematological assays were done as the samples came in, but only when the cohort was completed were these biochemical measures done. And the beauty of that was that you could use the cohort as its own control. So rather than measuring the people from the north of England, the middle of England, the south of England at different times, they were randomly measured. So the whole cohort was randomized, and then we measured it, so you know that there shouldn't be any assay drift. And then one of those kind of end of budget things that occur in the UK, we got a call saying, could you spend £20 million in a month? To which, of course, the answer is always yes. And then you work out how you do that. And it was down to particularly Peter Donnelly in Oxford bringing together a working group that developed, working with Affymetrix, an array, putting onto that array things that they thought were going to be important, and getting that genotyping done on all 500,000. And making those data available. And those data, I think, have helped people to identify a lot of associations between genes and disease, but also a PhD student at the Broad called Kira, working under Setka Thierson, had a very nice paper coming up with this idea of polygenic risk scores, of combining variants of small effect and identifying a few percent of the population that were at equivalent risk of heart disease to someone with familial hypercholesterolemia. And as you know, and as we'll see in this meeting, people have gone on to do more of this. This is work from a spin-out company that Peter Donnelly has set up called Genomics PLC, developing polygenic risk scores. But many other people have done this for different conditions, be it breast cancer, be it heart disease, be it prostate cancer. And one can imagine how this could be incorporated into clinical practice to help us to use screening more effectively and use preventive strategies more effectively. Then I got a call from Aris Barris at Regeneron saying, is it really possible to get access to the samples in UKBiobank? And I said, yes. So he pulled together a consortium, initially with GSK, then with other companies, and they funded the exome sequencing of the whole cohort and then made those data available to all researchers. And one example from work Regeneron have done is a druggable target for obesity. They found a variant in the exome associated with a substantial lower blood pressure. They confirmed it in a cohort in Mexico and also one in the US. And they, I think, now are working with Sanofi to see if they can develop a therapeutic against that. But again, all those data are available for people to try to identify targets for a range of different diseases. And then the Wellcome Trust particularly drove forward a public-private partnership with UKRI and with 4 companies to support whole genome sequencing. Matt Hurls from Sanger chaired a working group on behalf of UKBiobank to help guide the work from our perspective. And the Sanger and Decode in Iceland did all of the sequencing. And this graph just shows the sequencing that was going on against the targets throughout the pandemic. So despite the pandemic, they were creating the sequence data at the pace they were intending to do. And again, those data are starting to be made available. One of the interesting things that Carrie Stephenson at Decode has done has been to look at the genetic data, the sequence data, not just in the majority, the Caucasians in this study, but also in the kind of 2 main minority groups, and showed how even with 10,000 individuals, one can get some very powerful associations with this whole genome sequence data. So smaller proportion, but still powerful data there. And one of the discussions that's now going on in that working group that Matt Hurls has chaired, along with people like Matt Luce in the UK, is whether one could benefit further from doing long-read sequencing. One of the things that's really been, I think, particularly valuable in UKBiobank has been waiting until the technology got to the point where one could really do it at scale. And it looks as if now companies like Oxford Nanopore, but others, could do long-read sequencing at this sort of scale, which would help with phasing, but also might help in terms of looking at things like epigenetics, whatever that means. You'll explain that to me. But really to kind of understand the full genetic determinants of disease. So we think it's kind of getting to the point where that's possible. One of the things we've seen over the last 5 years with first the exome sequencing, then with the whole genome sequencing, is increasing number of both pharma and bioinformatics companies starting to see the value of supporting enhancements in UKBiobank that then benefit all research by all researchers, and we're hoping that that might be what occurs here. So again, work by Carrie Stephenson showing in small scale the benefits of long-read sequencing, this paper in Nature Genetics. Initially, when UKBiobank was started, the thought was that one would never be able to do these kinds of assays on 500,000 people. It'd be too costly. And so the strategy initially was case control. Wait until you had 10,000 breast cancer cases, pull the samples out from them, 10 or 20,000 controls, and compare those. The problem with that is it's great if you want to do stuff on breast cancer. But if you've done those assays on the breast cancer, and then sometime later you do the same assays on another set, you don't know that you can compare directly the assays done at different times. So ideally, one would do the assays all at the same time, as we did with the biochemistry, in a random order across all the cohort, so that you don't have any systematic shifts in the data. And so very rapidly, the International Scientific Advisory Board, which was initially chaired by Stephen McMahon from Sydney, then from, most recently by Bernard Keaveney from Manchester, said, hold on a second. You don't assay the sample, just wait. Just wait until you can do it on the whole cohort, because otherwise you'll deplete the cohort and you'll create data that you can't compare on the same assay. So by contrast, if we can get cohort-wide assays or random large subsets of the cohort done such that you can then do another random large subset until you have done the whole cohort, you avoid these problems. Much more cost effective. I can tell you, if you go out and you say you have £20 million and you need 500,000 people genotyped, you push down the price to a price that allows you to get 500,000 people genotyped at £20 million. So the ability to kind of push down and make, not just help Biobank, but then reset the cost of these assays, I think is a very powerful one. So very cost effective, minimizes depletion, improves the quality control, as I say, use the cohort as its own internal control, and it supports different comparisons different researchers want to do on different diseases. So case control is lower cost, it's quicker, but in the long run, it's much more cost effective to do these approaches if you can get the funding. So the good thing is we haven't depleted the sample. The vast majority of the sample, 15 years on, is still there waiting. And we're starting to see interest, particularly from pharma, but also in collaboration with charity and government, to do these assays. So Nightingale Health, who are here, have supported doing their metabolite assays on the whole cohort. A consortium set up by Chris Whelan at Biogen, bringing in more and more companies, supported initially the 1,500 protein platform BioLink, but then with more companies come in, they were actually able to go to the 3,000 protein platform on about 60,000 participants. And then there's some funding from the Chan Zuckerberg Initiative to Sanger to start to look at looking at RNA sequencing in samples that we're collecting. But our hope is over the next 5 years that this will really kind of escalate to doing more and more assays. We then thought, well, what more should we be doing in terms of assessing the participants in UK Biobank? We'd asked lots of questions, and we thought there were other questions we could ask, so we used web questionnaires to the 1/3 participants for whom we have email addresses to get more information about their diet, using diet diaries online, about their occupation over their lifetime to get about exposures. But the measurements we thought were a bit limited, blood pressure, height, weight, impedance, respiratory function, etc. And so we thought, well, what gives you information about people's body? Imaging. And we wrote to imaging experts around the UK and said, we want to image 100,000 people. And they said, you're really right. Imaging is so important, but surely you meant 10,000. Those were the emails we got back. And we said, no, I'm afraid not. So Paul Matthews brought together a working group. And within that working group, individuals with expertise in brain or heart brought together working groups in their area. And as again, hundreds of people across the UK were essentially involved in determining protocols, piloting those protocols, and putting together something that would allow us to do MR of the brain, the heart, the body, DEXA bone and joint, carotid ultrasound, and a repeat of most of the baseline assessment, including more detailed assessment of cognitive function and collection of biological samples. We've had to pause that with the pandemic at about 50,000. We've now resumed the imaging. And one of the questions we got when we went to the peer review was, if you create all this imaging data, how are people going to manage it? It's like providing a lot of samples with no assay. To which the answer is, if you build it, they will come. And what happened was creating the problem resulted in people producing the solution. So particularly in the brain imaging area, they started to work out automated approaches to analyzing the brain imaging, creating, if you like, assays of the brain imaging data, these image-derived variables that researchers could use, That's now happening in body imaging with a company called AMRA. Prospective is doing the same with the liver, and various academic groups are doing the same thing with the heart. So developing automated approaches that are then turning the imaging data in UK Biobank into usable variables, but also starting to be used in clinical practice. So stuff that's been developed in UK Biobank on heart image analysis is now being rolled out into practice by various groups. This is one example. There are lots of examples of the kind of different groups coming together. So this is genetics, imaging, and the statisticians. So they found 4 hits on this Manhattan plot, 4 variants that were associated with a hyperintensity in a particular part of the brain, indicative of microbleeds. When they looked at each of the hits, they were associated with a different part of that part in the brain, but all 4 of the hits were associated with iron transport and metabolism. And so Paul Matthews said, well, are these actually bleeds? Or is this something to do with iron deposition that is mimicking bleed? So is this identifying a potential target for cognitive decline that relates to iron transport? I mean, a hypothesis, but I think just an example of how the data, as we've seen with genetics, letting the data tell us what's going on rather than us telling the data what's going on, is an interesting way in which Biobank is helping to drive things. Because we had to pause the imaging project with UK Biobank, we thought, well, what do we do? Do we sack everybody and close down and mothball these imaging centres? So we have 4 imaging centres in the UK, in Newcastle, in Stockport, in Reading, and 1 in Bristol. Instead, what we did was we thought, well, we've got 50,000 people who've been imaged before the pandemic. With the support of UKIMRC and the Wellcome Trust, they said, well, rather than close down the centres and make everybody redundant, which would cost a lot of money, why don't you use that money to bring back some of these people who've been infected subsequently and match controls? So the idea was to generate a kind of unique resource where you have pre-infection imaging and post-infection imaging. So we brought back 1,000 people who'd been infected since their initial imaging and 1,000 controls, and made those data available. And again, a single example here from the brain imaging, looking at the first 400 infected and 400 controls, and they found differences in the infected versus the non-infected, particularly this time, the first wave, in the taste and smell areas of the brain, but not really anywhere else. And there are now cardiac studies that are coming out showing very little effect of the infection on cardiac function. We're now thinking about, well, what else? So there are discussions going on about, well, could we bring back a larger proportion of the image participants? So once we completed imaging of 100,000, our studies indicate that we can get about 60% of people to come back for repeat imaging. So could we get 60,000 of them to come back for repeat imaging, to add in optical coherence tomography, which was done at baseline, and additional types of sample? And this would allow the ability to look at changes in risk factors. So the impact of changes on health outcomes, changes in imaging measures on health outcomes, but also genetics and lifestyle and environment on changes in imaging measures. So we can see how that could be used in a range of different ways. And then that started us to think, well, why don't we bring back everybody so we can look at changes? Changes in biochemical measurements, changes in their cognitive function, changes in their aging measures. And again, we're kind of exploring that, as well as exploring the possibility, based on some work that we did on in the pandemic, around trying to recruit first-degree relatives. So the third aspect of UK Biobank is duration, 15 years of follow-up. We have information on death, cancer, hospitalization, and now primary care data during the pandemic under the COPI notice with the help of Martin Marshall and at the RCGP to get access to all of the GP data for COVID-related research that has been fantastically valuable. It really adds a huge amount, and we're hoping that we can continue to get access to that. In terms of the scale of the numbers of particular diseases, this gives you an idea of what we've seen up to 2020, but the numbers that are coming through in 2027 and 2032, increasingly powerful in terms of the ability to study an increasing range of diseases. We're looking to see whether we can link into particular health records for particular kind of health outcomes to get more detail, not just about the participants, but about their particular health outcome by increasing linkage, and doing some pilot studies about clinical imaging linkage or histopathology, looking at introducing smartphone apps to see whether participants would be willing to have those so we can assess them over time. And expanding the web-based questionnaires that we use, not just to assess exposures, but to assess health outcomes that are not detected through linkage to health records around health and well-being and sleep and cognitive function and mental health. The problem is you get all of this health record linkage, but how do you turn it into very specific and very accurate diagnoses? And so there are now kind of efforts going on about how at scale to combine different sources of information about health outcomes. And we're working particularly with people at the Turing, like Chris Holmes and Russ Altman at Stanford, to look at challenges that might be put out there for the informatics research community to help us to use AI and machine learning to develop automated approaches to determining specific diagnoses in individuals. So finally, the kind of fourth dimension of access. So there's no preferential or exclusive access. I'm the principal investigator only in the sense I don't do any investigation on UK Biobank. It's a resource for others to use. Researchers only have to pay for the cost of accessing the resource. We're trying, as I said, to limit the access to biological samples to ensure we don't deplete it. And the only requirement is to put the results into the public domain. So we've seen increasing numbers of researchers using it, and most interestingly in the last few years, an increase not just in Europe and North America, but in the last couple of years from China. And now we're looking to see how can we spread it out to LMIC countries and encourage use in those countries. Increasing numbers of publications, some 1,700 publications based on UK Biobank in 2021 alone in a range of different areas and journals, and increasing numbers of citations. The thing we also were able to do as part of the whole genome sequencing project with a grant from the Wellcome Trust was to go out for tender for a cloud-based platform. So instead of the data going to a researcher, researchers can go to the data, and all of the exome and whole genome sequencing are only available on the platform now. And that, I think, really democratizes access increasingly. You don't need a big computer to use UK Biobank. You can go to this big computer. And as part of that tendering process, Amazon, AWS, agreed not only to provide free storage to UK Biobank, but $500,000 worth of free compute every year to early career and lower middle income country researchers. And so we're really looking to see how we can enhance its use. So finally, the fourth dimension, accessibility. So it's scale, depth, duration, and accessibility. And I hope that with your help in telling us what more we should do to make UK Biobank even more valuable for what you want to do, the best is still to come. Thank you very much.